Hello, Bio 104 students. Welcome to Lecture 4, where we will be covering phylogeny and phylogenetic inference. Here's a set of terms that you'll be introduced to through the course of this lecture. And as always, these slides will be available for download on Canvas. And here's an outline of the topics that we're going to be covering in this lecture. And at the end of the lecture, I'll revisit each of these point by point for a brief discussion to round out the lecture. Now, as we learned, perhaps the most important idea introduced by Darwin in his book on the origin of species in 1859 is that all life on Earth shares common ancestry. And this is illustrated in the only figure in his book in 1859 where basically he's showing ancestor-descendant relationships. And you can see in this quote from the text, he calls it this great tree of life. And he mentions that the dead and broken branches are in the crust of the earth. These are the lineages that were around but are now extinct, recorded in the fossil record, and life that covers the surface of the earth are the descendants of that life that is found in the fossil record today. So beautifully put, but what Darwin is getting at is that if all life on Earth shares common ancestry, they share a genealogical relationship. And if there is a genealogical relationship, we should be able to infer those relationships. And that is the science of phylogenetics. Despite introducing this revolutionary idea, it was about 100 years after Darwin introduces the idea that there was the introduction of an objective, data-driven way of inferring phylogenetic relationships. Prior to the development of phylogenetic systematics, which starts in the 1930s and extends through to the late 1950s and into the 1970s, all phylogenetics was essentially based on expert opinion and authority, and that there was no way to take a data set and have two scientists essentially agree or come up with the same conclusions given the data that was being used to think about how organisms were related to each other. So this starts in the 1930s with Walter Zimmerman, who was working primarily with plants, and through the 1950s with Willy Hennig, who introduces the concept of phylogenetic systematics. And Willy Hennig was an entomologist. Okay, so the fundamental theory of phylogenetic systematics has a critical message, and that is phylogenetic relationships are not to be confused with similarity. The concept does not refer to what organisms look like or how similar they are to one another. Phylogenetic relatedness is entirely and only about the relative recency of common ancestry. Therefore, closely related species can be very different from each other in how they look or how they behave or their ecologies, and distantly related species can be quite similar to each other morphologically. So let's look at an example. And in the late 1970s, this example led some people to lose their damn minds. And they lost their minds over an exhibit in the public part of the Natural History Museum in London. And the exhibit had to do with the phylogenetic relationships as inferred from phylogenetic systematics, which was a new science at the time with regard to the lungfish, the salmon, and the cow. So the question is, given these three taxa, lungfish, salmon, and cow, which of the two are more closely related to each other? So if we think about lungfish, they share many overall similarities with the salmon than it does with the cow. And these similarities include having scales, having fins as opposed to limbs, having gills, and just a lot of traits that speak to their fishiness, so to speak. However, the lungfish has several shared derived traits with the cow that include lungs and a nasal passage connection between the nostrils and the mouth, called a choana. And indeed, if we look at the phylogenetic relationships, these are not defined by the overall similarity of the fishiness of the lungfish and the salmon, but by their evolutionary history. That is the relative recency of common ancestry. And that's it. In phylogenetic systematics, we're looking for the relative recency of common ancestry. So if we have a phylogeny of the major groups of vertebrates, we see that the lungfish is more closely related to the cow than either is to the salmon. Why is that? It's because the lungfish and the cow share a more recent common ancestor to each other than either does to the salmon. So here's the common ancestor of the lungfish and the cow, and here's the common ancestor of the salmon, lungfish, and cow. Now, phylogenetics goes beyond the esoteric study of how lineages are related in the great tree of life, but it has a lot of applications to human health and medicine. For example, by having a phylogeny inferred from genomic sequences of different viral lineages, we could see that HIV-1 and HIV-2 have two separate origins in terms of infecting humans from SIV-related simian viruses. That HIV has a common ancestry with chimpanzees, and HIV-2 has a common ancestry with a simian viral lineage that's found in Old World monkeys. Now, the application of phylogenetics goes beyond just understanding that HIV has multiple origins in terms of HIV strains that we find in humans. But if we go and we actually sequence those genomes through time, that is, when we have a viral sample that's isolated at a given time, it essentially timestamps that lineage. And so going all the way back to 1959, from a historical sample from an immunocompromised patient who died in Kinshasa in 1959, and extending, in this case, all the way up to about 1998 or 2000, what the scientists were able to do is trace the branch length to the common ancestor of HIV, the common ancestor of all HIV-1 that we find in human populations. Then you could take the branch length, the genomic branch length, how many changes have occurred in that viral lineage through time. So we can essentially treat the viruses in a sense as fossils. And we could not only see the rate that the viral genome is evolving, but we can ask, given this rate of change, can we estimate the branch length of the 1959 sample? And we see that here. Here is the box that contains all of these specimens and their branch lengths. And if we extrapolate out, we come very close to predicting the 1959 sample. In fact, the prediction is 1957 plus or minus 10 years. So feeling confident about being able to predict the 1959 sample, we can extrapolate out to the origin, and we can see that HIV-1 likely transferred from a common ancestor lineage that is infecting chimpanzees to humans at a time that extends from about midpoint of World War I to about midpoint of World War II. Clearly a time period of global upheaval. Okay, let's dig in and look at the parts of a phylogeny. So essentially, your lines here are the branches. Now, branches that are in the inner part of the phylogeny we call internal branches, and branches that extend out to the organisms that are in the analysis we call terminal branches. And the organisms that are represented here at the end of these terminal branches, we often call the tips of the phylogeny. Where the branches intersect, we call nodes. So a node that leads to the terminal branches, basically we can differentiate from nodes that are more internal in the tree. So an internal versus a terminal node. Going back, the branch that connects the common ancestor node we refer to as the root node, and all of the nodes essentially are inferred common ancestors. So we would say this node here represents the hypothetical common ancestor or most recent common ancestor, often abbreviated as MRCA, between the chimpanzee and the human. Now, this phylogeny is drawn where the branches correspond to the estimated timing of divergence among these lineages. Here on the x-axis, you can see this is shown in time in terms of millions of years ago. So the way you would read this phylogeny is that the chimpanzee and human share common ancestry at a time of approximately 5 million years ago. Now, with the phylogeny, it doesn't matter which taxon is listed on top or bottom. All that matters is that the common ancestor contains the same, or the descendants are the same taxa. So what I'm trying to say is that you could go ahead and rotate these branches where the human is on top instead of the chimpanzee, such as we have over here on the right. Basically, essentially, these two phylogenies are synonymous with each other. They are the same trees. What matters is you have a common ancestor that is subtending the human and the chimpanzee. The order that they're listed in the tree doesn't really have any meaning at all. Okay, so we're going to talk about 4 fundamental ideas in phylogenetics through the course of this lecture. And the first is that phylogenetic trees consist of monophyletic groups. A monophyletic group is a common ancestor in all of its descendants. However, to think about monophyly, we have to think about instances of groups that are not monophyletic. And then we have to think about the traits that we use to infer the phylogeny. So in this sense, we're going to be looking at terms that we're going to be using throughout our discussion of phylogenetics. And some of these terms we'll be using throughout the remainder of the Bio 104 course. So monophyly, as I said, is a common ancestor in all of its descendant taxa. Paraphyly is a common ancestor where for some reason, some of the descendant taxa are not included. Polyphyly is when you have multiple common ancestors, and I'll show you examples of this in a moment. A clade is another name for a monophyletic group. That is a common ancestor in all of its taxa. And we'll be using the term clade quite a bit through the remainder of the course. Homology we covered in the first lecture. Homology is just shared similarity due to common ancestry, and all shared derived character states that we're using to provide evidence for monophyly are by definition homologies. And homoplasy is similarity among lineages for any other reason beside common ancestry. Homoplasy is usually due to convergent evolution or evolutionary reversals, that is, loss of a traits, and similarities due to homoplasy are not homologies. Okay, so here we have a phylogeny of lineages encoded here with the letters A through F, and monophyly is a group where you have a common ancestor and all of the descendant species, all descendant lineages, okay? No lineage descended from the common ancestor is excluded from the group. Now, paraphyly is a group that does not contain all of the lineages that are descended from a single common ancestor. And a classic example of paraphyly is reptiles. So if you think of reptiles as a natural group consisting of lizards, snakes, turtles, and crocodilians, you're partially correct. And the way that reptilia is not a monophyletic group, it is because we're excluding birds. The fact that crocodilians and birds share common ancestry relative to all these other reptiles indicates we have a common ancestor and we're excluding one of the descendant lineages, in this case, birds. So therefore, reptiles are paraphyletic. Now, this can be easily fixed just by including birds as reptiles. Now, polyphyly is when you have multiple common ancestors that are separated by other common ancestors. And polyphyly really is, in a sense, a mistake of classification or taxonomy. So what would be a good example of a polyphyletic group? Well, what if we wanted to make a taxonomic group that included all flying vertebrates? So we would have pterodactyls in there, we would have birds, we would have bats, and clearly these groups are all distantly related from each other. That would be a polyphyletic group. All right, so I'm going to give you a moment to think and challenge you with some tasks in reading phylogenies. Now, normally we would be taking a stretch at this time. You'd be talking to those of you in your adjacent areas in lecture. But since we're doing this all online, we're going to take a slightly different approach. So what I want you to do is think for a moment and jot down what are the different kinds of groups in green, orange, and blue. And then how many monophyletic groups are there in this phylogeny? Okay. So what type of group do we have in the green box? That's right, paraphyletic group, because we have a common ancestor, but we're excluding some of the descendant taxa. What kind of group do we have in orange? Right, polyphyly, because we have multiple common ancestors that are separated by other common ancestors. And what's the group that we have in blue? This is the easy one. Right, it's a monophyletic group because we have a common ancestor and all of its descendant taxa. Now, how many monophyletic groups are in this phylogeny? Well, the way I think about this is I usually start from the top or the bottom of the tree and move in the other direction. So let's start from the bottom. So we have a common ancestor and the descendant taxa. That's one group, two monophyletic groups. Here's the third monophyletic group. The whole group is the fourth monophyletic group. This clade here is the fifth monophyletic group. Here we have the sixth monophyletic group, the seventh monophyletic group, the eighth, I'm sorry, I double counted one. Let's do this again. There are 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 monophyletic groups. Let's do that one more time. 1, 2, 3, 4, 5. The whole one is 6, 7, 8, 9. 9 monophyletic groups. Okay. The next fundamental idea in phylogenetics we're going to talk about is that shared derived character states are what provide evidence to the composition of monophyletic groups. Okay? Shared derived character states. So I'm showing you here is a phylogeny of vertebrates. And here we have the tetrapods, which are defined by having 4 limbs. Here is a group of tetrapod vertebrates known as the amniotes. They're characterized in part by having an amniotic egg. And within the amniotes, we have birds, crocodilians, snakes, lizards, turtles. We also have us, the mammals. And mammals are defined by having hair and lactation in part. Okay? So mammals are defined by having lactation and hair. Even though mammals have an amniotic egg and have 4 limbs, mammals are not defined by having 4 limbs and an amniotic egg. They are defined by the shared derived traits of hair and lactation. The ancestral character states of having an amniotic egg and 4 limbs, while present in mammals, they're not informative about the monophyly of mammals. Therefore, they're not traits that define mammals as a monophyletic group. The next fundamental idea is, in a way, the way that we provide polarity, the direction of evolution that we're inferring in the phylogeny, as well as the character states in the phylogeny, the shared derived character states. And this is called outgroup comparison. And in a nutshell, what we're doing with outgroup comparison is we're looking at the character states of the group we're interested in inferring the phylogeny, then bringing in knowledge about the character states in groups that are closely related to the group of which we're trying to infer the phylogeny. So the challenge we have is if we're scoring phenotypic traits, a binary character, in this case, scored as 1 or 0, or in the case of nucleotide states, there's only 4 states a nucleotide site can be, right? A, T, G, or C. What we want to know is the direction of evolution going from character state 1 to 0, or 0 to 1, or A to G, or G to A. How do we determine this? Well, we determine this through outgroup rooting. So let's use an example very close to home. And let's think about the presence of tails in primates. So our in-group is the group of which we're interested in inferring the phylogeny, in this case, primates. And the outgroup will provide evidence of character state polarity, that is the direction of evolutionary change. We want to know, what is the derived state for primates? Is it having a tail like we see in this old world monkey? Or is it being tailless like we see in the chimpanzee, our fellow great ape? Okay, so if we have a phylogeny of primates, we have a bunch of traits that define primates. However, we have a node here that represents the common ancestor of primates, and the node beyond that is unresolved. We have no knowledge of the phylogenetic relationships of the lemurs and monkeys and the great apes. And these are scored with having a tail as a T or no tail as an N. If tails are derived, then the presence of tails is a shared derived state that provides evidence of monophyly of lemurs and monkeys. However, if losing an absence of a tail, the loss of the tail is a shared derived character, it will then provide evidence for the monophyly of the great apes. So how do we figure this out? Well, we have to include some outgroup lineages that are outside of primates but closely related to primates. So we could reach out and look at these other lineages of placental mammals. In the in-group character state that is present in the outgroup is most likely to be the ancestral character state for the in-group. So you can see all of these mammals have tails. I think you could then guess the direction that this conversation is going. So by outgroup comparison, here are the outgroups, they have tails. Here are our primates with tails. We would argue that the most recent common ancestor of primates had a tail. So if no tails are derived, then the absence of a tail is a shared derived character state that provides evidence for the monophyly of the great apes. However, presence of tails is not evidence of monophyly of the old world monkeys, because this is an ancestral character state that is present in the in-group, okay? Because the outgroup has tails, we're inferring that having tails is ancestral for primates as a whole. However, losing tails provides evidence for monophyly of great apes within primates. Now, these outgroups provide a root to the phylogeny. So what I'm showing you here are 2 identical unrooted phylogenies. However, in these phylogenies, if the outgroup resolves on a different branch of the in-group, and we go ahead and we pull on that outgroup here, pull on this red part of the root, we then polarize these phylogenies. And you can see that even though the unrooted trees are identical, with a differing resolution of the outgroup, we get very different phylogenetic trees that result after rooting. Okay, the last fundamental idea that we're going to talk about in phylogenetics, it's going to take up the majority of our time, is how do we pick the optimal tree? So we have character data. This could be from phenotype, or more likely, as in the early 21st century, DNA sequence data from gene and or genome sequence data sets. What is the method that we use to find what we think is the optimal phylogeny? That is, what phylogeny best explains the character variation that we're observing? Now, the very early days of phylogenetic systematics, the principle of parsimony was the way of finding the optimal tree. That is, the phylogeny that required the fewest number of changes among the characters was considered the optimal phylogeny. Now, parsimony is still a powerful tool for inferring phylogeny and is a great teaching tool as an introduction to phylogenetics. But as we're going to learn, there are ways that evolution can fake out parsimony. And now we have more sophisticated statistical approaches that take into account other lines of information that we use to find that optimal phylogenetic tree that best explains the data. That is, the phenotype and or molecular data that we're using to infer phylogeny. Okay, so this is the problem. How do we infer a correct or optimal tree from the character matrix? Now, let me just take a moment and say the correct tree, I mean the optimal tree. Here's the secret about phylogenetics. We'll never know if we're right. We're reconstructing the past. We're making an inference. There's no experiment that we can do to prove that one phylogeny is more optimal than the other, all right? So when I say the correct tree, in a sense, I'm thinking about in a model simulated world, we know this is the correct tree. How do we find, how do we infer that data? How do we infer that tree given our character matrix? Okay, this is the way character matrices look. The taxa, the species or lineages that we're inferring the phylogeny for are here in the rows. And the columns represent the characters, and the ones and zeros could be 0, 1, 2, AGTC. The varying traits here, whether 0, 1, or AGTCs, represents the different states at that character. All right, so these are called the character states. The whole column is the individual character. So characters and character states are in the columns. Species or taxa for which we're inferring the phylogeny are in the rows. All right. Now, the way we do this is we just march through. We say, okay, character 1 has character state 1 in taxa B and C, and character state 0 and A. So here we infer a change of character 1 from 0 to 1. And for character 2, we see that there's a change from 0 to 1 leading to taxa C, and the same for character 3. So the way we figure this out is by mapping, physically mapping these character state changes onto our differing trees. The differing trees are the alternative hypotheses, all right? Different phylogenies are alternative hypotheses, and the hypothesis that we're looking for the hypothesis that best explains the data. And using parsimony, that is the tree that requires the fewest number of character state changes. All right, so what will you do when the characters are in conflict? When they appear to be telling different stories. Okay, so here's the data set we just looked at. There was no conflict. All the characters agree. But here you could see character 3 conflicts with both character 1 and 2. Character 3 is arguing for common ancestry of A and B, whereas characters 1 and 2 are arguing for common ancestry of taxa B and C. What do we do with this? Well, we use parsimony. That is the tree that will require the fewest number of changes as a way of determining the optimal phylogeny. And sometimes characters are going to conflict with each other. More often than not, there is going to be character conflict. This character conflict is known as homoplasy. These are differences that are due to processes other than homology, all right? So let's look at our model tree here, and let's complicate this a bit. We have 5 taxa and 5 characters. And remember, we're scoring the different character states in the columns, and the rows are the character states that are scored for each of the taxa, right? Okay, so let's look at character 1. This is very straightforward. We have a character state change from 0 to 1, leading to taxa C and D. Character 2, It has a character state change leading to taxa A and B. Character 3 provides evidence for common ancestry of taxa A, B, C, and D. Now, character 4, interestingly, on this tree, basically has some conflict. The character state must either evolve convergently twice, that is along the lineage leading to B, the lineage leading to C, or evolve once and be lost twice. So here, if it evolves twice, that's 2 steps. Here, if we say it evolved once and was lost twice, that's 3 steps. Therefore, we prefer the optimization here where it evolved twice, as opposed to having 3 evolutionary steps where it evolved once, was lost twice, okay? Then character 5 is another character that is can be thought of as providing evidence for common ancestry of a shared derived character for the clade composed of species A and B. Okay, now this tree requires a total of 6 evolutionary steps for these 5 characters. Remember, every character had a single evolutionary step, except for character 4 that had homoplasy, right, on lineages B and C. But if we go ahead and just count the number of changes, this tree requires only 6 changes, all right, for the 5 characters. Now let's think about an alternative phylogenetic tree. So we could say, well, maybe these traits that were changing independently on B and C are evidence for common ancestry of B and C. So let's think about a phylogeny that relates B and C as a monophyletic group. Let's do that. If we look at this tree, this alternative tree has 8 evolutionary steps for the 5 characters. This is not the most parsimonious tree. The most parsimonious tree is preferred and is considered optimal and is considered a better explanation for the data and the variation of the data that we're observing. So, using the principle of parsimony, by mapping these traits on the alternative phylogenies, we can see this tree requires 6 changes. It's the most parsimonious tree. It's considered the optimal hypothesis. We can reject this hypothesis on the right because it requires 8 evolutionary changes. Now, this is a very simplistic approach to thinking about phylogeny reconstruction. However, this is how it's done. So if we're looking at hundreds of species, as we're going to see, we're going to engage with some of the largest numbers that you will ever engage with in your scientific life as a student or a practicing scientist. I'm not exaggerating. And with millions of base pairs of genomic data, computer algorithms are essentially figuring out the number of changes on the trees. That is, if you're using parsimony. So while this is a straightforward, simple example, if you ramp this up orders of magnitude with regard to the number of taxa, as well as the number of characters and character states that we're dealing with, this is the logic and the method behind inferring phylogenetic relationships using parsimony. Okay, now we've got the trait matrix and we know how to map changes on the tree. And so we want to find the simplest tree. Now, one possibility is we could just evaluate every possible tree. So if there are 3 species in the phylogeny, there are 3 possible phylogenetic trees. If there are 4 species in the phylogeny, there are 15 possible trees. And maybe we could go ahead and map the changes for our matrix on every possible tree and be assured that we have found the optimal tree because we've looked at every tree. We've done what we would call an exhaustive search. However, the numbers get amazingly large. So if you look at the number of branching phylogenies that are rooted, as you increase the number of taxa in your analysis, it gets enormous. So even if we jump from 4 species to 10 species, we would have to evaluate 34.5 million different phylogenetic trees in an exhaustive search. Even if we double that, we go to 20, this is the number of unique phylogenies we would have to evaluate. Now, if we're looking at 500 species, that is 10% of the species diversity of living mammals. We have a number that makes Avogadro's number look tiny. This number is huge. And we're talking about 500 species. And for comparison, the universe contains only about 10 to the 89th power number of protons and has an age of about 5 times 10 to the 17 seconds, or 5 times 10 to the 29 picoseconds. So biologists go out and brag that we, as evolutionary biologists, are dealing with numbers that are far larger than any that are encountered in the physical sciences. But the point is, this is an enormous number of trees to evaluate, and there is no way to exhaustively evaluate them all. How do we do that? We do that by using algorithms that we call heuristic methods and branch swapping. These algorithms take these more complicated phylogenetic problems and break them up into smaller problems and bring them back together. And by doing that, one of the benefits of these methods, it allows the algorithm to eliminate whole ginormous sets of less than optimal trees. And so by eliminating all those trees, we could basically take what is an incredibly large problem with regard to the number of phylogenies that have to be evaluated and really distill it down to a much more manageable number of trees. Okay, now parsimony is wonderful in that it really provided a great logic for thinking about how do we discriminate among alternative phylogenetic hypotheses. Now, the problem with parsimony is when we were doing that exercise with the character mapping, we didn't really care how many changes were on one branch versus another. Parsimony is completely agnostic to where in the phylogeny the changes are occurring. We could think of branch lengths. So in the HIV example, remember we were using the branch length from the time of the sampling of the viral lineage to the common ancestor as a piece of data. And that branch length reflects how much genetic change happened genomically in those viral lineages, okay? Now, one thing that we know by studying biodiversity is that some lineages have varying rates of evolution. Not just molecular genetic evolution, but even phenotypic evolution. This is a problem, a trap that parsimony falls into, because parsimony is completely agnostic to the amount of evolution happening in a lineage, all right? Now let's look at this phylogeny here. This phylogeny looks a little weird. That's because the branches are scaled to reflect the amount of change that is observed in the characters used to infer the phylogeny. But the true tree here is lineage A and lineage B share common ancestry, and lineage D and lineage C share common ancestry. So A and B and C and D are two monophyletic groups or clades. And what we could see is that on the branches leading to lineage A and lineage D, we have a lot of character evolution. And just by chance, where I've stuck these green dots, just by chance, they're converging on the same character state. So think if these were nucleotide sequence characters, there's only 3 other states that a nucleotide character state can evolve to. So if it's at a G, it can only go to a T, a C, or an A, right? So just think about this. If there's a lot of change happening in a lot of the sites in DNA sequence, just by chance, a site can convergently evolve to the same nucleotide character state. So just by chance, because they're evolving at a high rate, they will independently evolve the same character states. And when you do this, when this happens and you use parsimony, you're going to infer the wrong tree. So even though this is the true tree, we will infer that lineages B and C here, the short branches, are a monophyletic group, whereas A and D, the long branches, are a monophyletic group. This is the wrong tree, and A and D are inferred as a monophyletic group because of long branch attractions. That is basically convergent evolution in those lineages fakes us out. And we think they're homologies, we think they're shared derived character states, we then infer the wrong tree. Now, the problem with this is you can't just go out and collect some more data. The problem is that long branch attraction becomes positively misleading, meaning that as you add more data to the problem, you keep inferring the wrong tree. How do we get around this? Well, we get around this by understanding that parsimony doesn't take into account unobserved changes in character states and the branch lengths in the phylogeny. The longer a branch length in the phylogeny, maybe we can infer that there's going to be a higher rate of change for any given character we're looking at. Whereas a short branch, we would infer, or we would guess that there's going to be a slower rate of evolution in that lineage. If there was a way to incorporate that information into our phylogeny reconstruction, that would be really helpful. And what I mean by unobserved changes is illustrated here with this hypothetical evolution of 2 DNA sequences from a common ancestral DNA sequence. So here's the ancestral DNA sequence. It diverges, let's say 2 lineages diverge in allopatry. And what we can see here is unobserved changes are those that are in red. So even at this site, even though they're going to be ultimately T and A, T in one lineage, A in the other, in this lineage, it actually went from A to C to T, okay? And the changes that are consistent, meaning there are no unobserved changes, are in blue. You can see the majority of changes are unobserved. Majority of changes are unobserved. So what we want is a way, a probabilistic approach to estimate the amount of unobserved changes in these traits. And this led to, about 50 years ago, the introduction of models of DNA sequence evolution. And the 2 models we're going to talk about in this course are the Jukes-Kanner model here on your left and the Chimera 2-parameter model on the right. Now, all these models are doing is they're trying to take into account what is, or trying to estimate what is the probability of having an unobserved change. Now, the Jukes-Kanner model on the left is the simplest molecular evolutionary model. It's arguing that nucleotides occur at equal frequency and that any change between any of the 4 states is equiprobable. So going from A to G has the same probability of changing from A to C, all right? And any of these changes are equiprobable. It's just that there's a probability of mu over 3 for a change from 1 to any of the other 3 bases, all right? So that probability allows us to estimate how much unobserved changes there have been. Okay, on the right, we have the Chimera 2-parameter model that incorporates a little more of the natural history of how we know DNA sequences evolve. So going back to biochemistry, you probably learned that there are 2 chemical classes of nucleotides. There are purines, which are adenine and guanine, and then there are pyrimidines, which is cytosine and thymine. Now, if we look at how DNA sequences evolve, what we see is that there's a higher rate of changes of substitutions within chemical classes, meaning that purines tend to change to other purines, pyrimidines tend to change to other pyrimidines. Changes within the chemical class are called transitions. So changes between A and G and C and T are transitions. Transversions are changes between the chemical classes. So a change between a purine and a pyrimidine is a transversion. A to C is a transversion. C to G is a transversion, okay? So the fact that transitions occur more frequently than transversions, we could take that information, and that's exactly what the Chimera 2-parameter model is doing. It says there's an inequality of rates between transitions and transversions, and there's a probability of alpha for a transition and beta for a transversion change. And these are models, and then there are more complicated models that we're not going to be talking about in this class that are used to take into account the unobserved changes. Also, coupled with branch lengths, the longer a branch in a phylogeny, meaning the more changes that are being reconstructed on that branch, the more likely there will be other changes. Therefore, we want to take that into account in reconstructing the phylogeny as well as estimating the unobserved changes. So moving beyond parsimony, I'm just going to mention this. We're not going to really get into the weeds with it like we did with parsimony. And for those who are interested in phylogenetics, there are upper level courses in ecology and evolutionary biology where you can really dive into this. But we'll think about maximum likelihood. Maximum likelihood is a statistical approach for optimizing a probability. Now, before, the ingredients that we were using to infer phylogeny from parsimony was simply the phylogeny and the data, right? So we're comparing alternative hypotheses, different phylogenies, and seeing which one required the fewest number of changes from our data. But now with these statistical maximum likelihood approaches, we're comparing alternative trees, right? Like this group of mammals here and their outgroup, but we're also taking into account the branch lengths. The branch lengths are going to enter into the probability of finding the optimal tree, and will include the model of DNA sequence evolution, such as the Jukes-Cantor model or Chimera two-parameter model that we just talked about. Then we will utilize the data, the model of sequence evolution, the branch lengths, to determine the optimal phylogeny. Okay, so in this discussion of phylogeny and phylogenetic inference, we covered quite a bit of ground in a fairly short amount of time. So remember, phylogeny represents Darwin's fundamental idea, perhaps the most important idea in The Origin of Species, that all life on Earth shares common ancestry. That phylogenetic systematics is fairly new to evolutionary biology relative to when Darwin introduced this idea. I would say roughly about 100 years after Darwin's book was published. You should think about the parts of a phylogenetic tree, how to read a phylogenetic tree, think about monophyly and those related concepts. You should know how to infer a phylogeny from information on character state differences. In fact, this will be part of one of your P sets. And be able to articulate how overall similarity can fail to infer the correct phylogeny. Remember, evolutionary derived traits are the basis for inferring relationships. This is a learned skill, but also philosophically, this is the logic of phylogenetic systematics. Groups are defined by common ancestry, and evidence of common ancestry comes from shared derived traits. You should be able to think about using outgroup comparison to determine character state polarity, as well as rooting the phylogeny. You should think about being able to use parsimony to reconstruct character state evolution, and using parsimony to identify the preferred tree when there's character state conflict or homoplasy. You should know when and why parsimony infers the wrong tree. In the case we talked about in this lecture is long branch attraction. And then think about model-based approaches of inferring phylogeny, maximum likelihood, but more specifically, be able to think about models of DNA sequence evolution, such as the Jukes-Cantor and Chimera two-parameter model. Okay, this is all we have in the lecture for lecture 4, covering phylogeny and phylogeny inference. Our next lecture will be an introduction to ecology. So until then, be well and be safe, and I look forward to hearing from you soon.